PGAs, which is PGLs. Last argument does the same thing. And so we should end up with our 6 PLs. How many of those PLs will we use to regenerate 3 RBPs? Okay, look right. That would be 15 carbons that we need to regenerate our starting material again. And so again, we ended up with one extra PL. 1 out of 6 is an extra one. And now we have 6 carbons. The extra carbon is the one from the first PL we made. And now there's another extra PL we made. Well, 6 carbons together will be joined together, and that forms a new glucose. Enzyme that just links those 2 PLs together. And there's that glucose. And so it's kind of a complicated system. You have to go around a few times. But eventually, you end up making 2 extra PLs, and those 2 extra PLs are linked together, and we get a new glucose. Then the whole process will keep going around and around and around, and it essentially turns into a glucose factory, making glucose from the carbon dioxides that are entering the alkyl cycle. What the glucose formula, main thing, of course, is cellular energy. All cells, including plant cells, love to use the glucose as their fuel source. So it's going too far. So that's the main point of the glucose. The plants like to store up the glucose as starch. There we go. They're not showing it on this diagram, but the plants also use the glucose to make cellulose, which is the fibers building material to make new wood and new parts of the plant's body. Any questions on it up to this point? All right. Fascinating process. When I was a graduate student at UC Santa Cruz, when you first enter graduate school, they make you rotate through several different research laboratories just so you get a taste of different researchers' research and to see which one you want to do your thesis work in. And then rotated through a lab and one who studied Rubisco from pine tree. I don't know why, a pine tree in particular. And it didn't really grab my attention at that point, so I didn't end up staying in that lab. But looking back on it, kind of a neat enzyme is definitely doing something important as part of the photosynthesis process. Anyway, any Calvin cycle questions? Okay, well, so notice that the Calvin cycle, as I've been showing to you so far, the first stable molecule that the carbon dioxide gets fixed into is PGA. This does not count as a stable molecule. And so for that reason, they call plants that do the Calvin cycle in this way that I've been showing you C3 plants, because CO2 first gets fixed as a C3 molecule. The reason why I want to point out this to you, all these types of plants, C3 plants, is there's a variation on what I'm showing here in different types of plants. And these other types of plants are called C4 plants. But first, let me talk a little bit more about the C3 plants, and then I'll show you the variation. This is supposed to be a leaf of a C3 plant. This is the top of the leaf right here. The cells at the top of the leaf. This is the cells at the bottom of the leaf. And review question: the cells in the middle of the leaf, where the photosynthesis takes place, are the blood cells, mesocyl cells, right? So this big green box right here is a mesocyl cell. And so inside the chloroplast of the mesocyl cells, where the Calvin cycle takes place. Here's a little opening where white cells. The stoma is the opening, but the cells that can open and close it. Remember that? Guard cells. They can get bigger to shut off the stoma, or they can shrink themselves to open up the stoma. Here's the stuff you see. Okay. And, well, don't peek at the black one, see if you remember. What's the name of this enzyme right here that grabs the carbon dioxide and grabs the Rubisco? It's called Rubisco is the enzyme, and it grabs the RUBs and carbon dioxide. Okay. So, watch it in action. Let's say that it's not a hot day. Moderate means not excessively hot, so it's a relatively not hot day. The carbon dioxides drift into through the open stoma. And so you get a nice high carbon dioxide concentration inside the leaf. And that's good because then the Rubisco enzyme is able to grab those carbon dioxides and fix them, feed them into the Calvin cycle. There they are. Now, under moderate temperature, C3 plants work just fine. The problem is when you get a hot day, when you get really hot weather, what happens is water can start evaporating out of the stoma. So now it's a super hot day. The inside of the leaf is a very watery environment because, remember, water is one of the ingredients for doing photosynthesis. You need water just as much as you need carbon dioxide for photosynthesis. And on hot days, water evaporates a lot. And so C3 plants have a problem. On hot days, water vapor can evaporate and escape through the stoma. The main purpose of the stoma is to allow carbon dioxide to come in, but certainly water vapor can exit through that same hole. And so on really hot days, C3 plants are in danger of getting dehydrated, and they might potentially get so dehydrated they can die. And so even though they don't want to, they get the guard cells to close the stoma. The guard cells essentially kind of swell up. The guard cells usually don't completely close the stoma. There's going to be a little bit of gap there, but it's not a big gap. And so you'll end up with a very low carbon dioxide concentration. A very small amount of carbon dioxide molecules can get inside there. And that's going to make for a very inefficient photosynthesis, which is a very low carbon dioxide concentration. You're just not going to have too many carbon dioxides available for that Rubisco enzyme to fix. And to make things even worse, Rubisco doesn't really have a good binding site for carbon dioxide. In other words, this binding site that grabs carbon dioxide with is not very well shaped, so to speak, to grab carbon dioxide. If it was a football player, it would be a football player that dropped a football a lot of the time. So even that low concentration of carbon dioxide can't get into you very effectively because the Rubisco enzyme can't grab it very efficiently. And you know, we're not supposed to project our human feelings onto non-human things, but I can't help but thinking the C3 plants must be frustrated in a situation like this because it's a nice, bright, sunny day. A lot of hot days are really sunny days, and so there's just tons of light energy falling on the plant, and it probably just wishes it could use that to do a lot of photosynthesis, but it can't because it has to close off the stoma, so dehydrate itself to death, and so that limits the amount of carbon dioxide that can come in, so it just can't do a lot of photosynthesis. Okay, so that's a problem for C3 plants. They work great at moderate temperatures, but at higher temperatures, their photosynthesis rate drops way low because they have to close their stoma, or at least most of them close their stoma. Most of the plants in our region of the world are C3 plants because although we have a few hot days every year, we don't sunny, but it's not really too hot. So our plants do pretty well. But if you think about the Earth as a whole, if you, where are we? We're somewhere around here, I guess. The further south you start heading, you get towards the tropical regions. And it gets hotter and hotter. So the plants in the tropical regions of the world are going to be constantly faced with really hot, sunny days. And so they have adapted a variation on photosynthesis that is going to allow them to do photosynthesis efficiently, even on very hot, sunny days. And this type of plant is called a C4 plant because this variation that I tell you about involves fixing the carbon dioxide not as a C3 molecule first, but as a different molecule that is a C4 molecule. All right, so let's see how these C4 plants do. When the weather gets hot, in the C4 plant, you still have to close your stoma. So the guard cells still get bigger, so you get a very, very tiny gap in the stoma. So you still end up with a low carbon dioxide concentration inside the leaf. That part hasn't changed. But what has changed is you fired Rubisco, so to speak. You don't use Rubisco as your carbon dioxide fixing enzyme anymore. In other words, this enzyme right here is no longer going to be used to fix the carbon dioxide. Why not? Because remember, it doesn't work very well with low concentrations of carbon dioxide, which doesn't find carbon dioxide efficiently. So in the C4 plants, they have another cycle called the C4 cycle that operates ahead of the Calvin cycle, before the Calvin cycle. And all the C4 cycle does is fix the carbon dioxide into a C4 molecule. It sort of prefixes the carbon dioxide before it gets fed into the Calvin cycle. Okay, so what does that mean as well? This enzyme right here, the C4 cycle, that fixes carbon dioxide, has a much better binding site for carbon dioxide. So even at the low CO2 concentrations that you get inside a almost closed off leaf, even at those low CO2 concentrations, this C4 carbon fixing enzyme still does great to store rapid CO2 very efficiently. So let's see. C4 cycle for the Calvin cycle and CO2 fixing enzyme of the C4 cycle, that enzyme right there, can fix CO2 at very low concentrations and still close the leaf. And so we're going to see that here, that C4 carbon fixing enzyme grabs the carbon dioxide, it becomes part of the C4 molecule like that. And then now that the carbon dioxide is fixed in the C4 molecule, that C4 molecule, since the carbon dioxide is already fixed, can be fed right into the Calvin cycle, and the Calvin cycle itself doesn't have to do with the carbon fixing. It's just essentially feed right into the Calvin cycle. So, certainly in hot weather, it's better to be a C4 plant because you can fix your carbon dioxide much more efficiently under the low CO2 conditions inside a closed off leaf. But it seems like every advantage has a disadvantage that goes with it. And the C4 plants pay a price for that. And here's what the price is. They have to use an ATP as part of the C4 cycle. In other words, they have to expend a little energy to do that prefixing of the carbon dioxide over here in this step right here. Whereas the Rubisco enzyme, that's part of the Calvin cycle, does not require ATP to fix the carbon dioxide. So they do have to expend an extra ATP, that's the cost, but they do get a benefit of being able to do photosynthesis on hot days. So, which is it better to be, a C4 plant or a C3 plant? Well, they both have advantages and disadvantages, and it really comes down to what part of the world you're in. If you're in a part of the world where it's constantly hot, the advantage of being able to do photosynthesis on really hot days outweighs the disadvantage of having to expend an ATP to fix the carbon, so you do better as a C4 plant. But if you're in a moderate temperature part of the world, where there's very few hot days, the cost of expending an ATP every time you fix a CO2 is too high, and you're better off just being a C3 plant and letting the visco enzyme fix your CO2s. Yes.
I know specifically, like, in between plants, different plants are going to create different amounts of glucose, right?
Yeah, I guess so, yeah.
At different rates, but between C4 and C3, which one creates the faster rate of glucose?
Well, again, I have to say, it depends on the temperature. Under hot conditions, the C4 plants would do it much more better, just because they're able to fix carbon dioxide under the low CO2 conditions that we find on hot days. And a C3 plant would do very little photosynthesis at all on a hot day like this.
So about within their respective desired climates?
I see what you're saying. Like C4 plants on hot days versus C3 plants on moderate temperature days. That's a good question. I don't really know the answer.